Okay, hello everybody, and welcome to this month's ASPB PUI Playbook office hours, faculty office hours. I'm sure most all of you are aware of the PUI section of ASPB. There's a little info on the screen that you can take a look at, ways that you can connect with us on social media, ways that you can see past meeting recordings on YouTube that are available for the whole community, and of course, connect with us on Plante. All right, so my name is Britt Moss, and I'm a new member in my first year on the PUI steering committee. And I'm a faculty member at Whitman College in Walla Walla, Washington. So this is the sort of south central part of the state. And our college has about 1,600 students, all undergraduate. And what I'm going to sort of try to lead some discussion today on is tips for designing a cure, a course-based undergraduate research experience. So I'm going to tell you just a little bit about some of my own experiences teaching cures, but I know we've got other folks in the Zoom room who I know have experience doing this, who can chime in as we enter discussion sort of afterwards. Okay, all right, so just real briefly, if you're not super familiar with this term, what is a cure? So it's a course-based undergraduate research experience. And so the idea here is to incorporate authentic research projects embedded in a structured laboratory class. I'm going to point you towards a resource that just came out last year. So Martinez, Vaz, and Bell in 2025 through the ACS Symposium Series published this book that's available that's all about cures. Anything and everything that you might have questions about or want to know, you can find excellent resources here. So I pulled that sort of definition of a cure from that book. So what are some of the benefits of cures for students in particular? Shown to improve student retention in STEM, especially if students are participating in an authentic research experience within their first couple years in college. Increased persistence and likelihood of students continuing on to graduate or professional school training. And can be especially impactful for historically marginalized students finding belonging and identity in the laboratory. But there's also, I think, benefits for instructors for teaching these types of courses. So it's a means of continuing to advance your own research program if you're integrating your own research into the CURE experience. I find a lot of joy in teaching the authentic process of science sort of alongside students. And I have found, right, that especially being able to do this kind of early to mid career for students in college, it prepares them to be better research students. So I've often found this as an opportunity to recruit students to come and do research in my lab for the summer who have taken these types of courses and are coming in better prepared to launch their summer research projects. Okay, so a couple of the types of cures that I have some experience with teaching here can really range. I've taught them both at the intro level and in upper level courses. So you can teach them as, for example, a full course. So I've done this with a class that I created called Synthetic Cell Biology. So I'm going to talk in the next few slides in more detail about that. So I'm not going to say a ton at the moment. But I've designed that class based around my own research projects. You could also do it just as a standalone lab, sort of accompanying an existing lecture course. I actually did that last semester for molecular biology. So I was teaching to cover for a colleague who was on sabbatical. I was teaching molecular biology lecture and lab. So I decided to make the lab kind of a modified version of my synthetic cell biology course. So I sort of adapted it for that purpose. And then another option is to develop a module that could go within an existing lab course. So this is something that we actually just did in our revamped introductory biology series. So in the second class of that series, which has a cell and molecular biology focus, some colleagues and I developed a 4 to 5 week mini project that happens at the end of that lab course. And it's a bacterial promoter mutation analysis lab that's been really fun. And this one was adapted from an already published cure. Ekdahl and Campbell published this on CourseSource. That's how I found it. Thought it was really cool. Adapted it to fit within the context of sort of our course here. So that was something I wanted to actually point out, is that there's actually a lot of already published examples of cures that you can use, right? Either to plug right into or to adapt to utilize in your particular courses. So I put a couple of those resource examples up here: CureNet, CourseSource, as well as examples of cures being published in journals like JMBE and Life Sciences Education. So those can be great resources as well. You don't have to reinvent the wheel necessarily. Okay, so I'll tell you just a little bit as an example about the synthetic cell biology course that I developed that's a full course cure. And so it's a 4-credit class. And it was designed really, my idea, my goal was to give students a real authentic experience of what it means to do science, right? So what does it mean to do science, right? It means hands-on lab work, right? So they were in the lab doing a semester-long research project, right? It means sharing out your results. And so the students did research posters at the end of the course to share out their work. We did journal clubs throughout the course of the class, so that students were reading primary research literature and having experience presenting and critiquing that work. And then we also talked a lot about how do you actually get your research funded. And I'll give you some examples of some of the ways that we have done that. But since I've been at Whitman about 10 years, I've had about 125 students who have participated in various iterations of this class and worked on these research projects. Okay, so the learning goals that I sort of set out for this class, one was it was going to be a survey of the field of synthetic biology, right? And this was going to be primarily through engaging with primary research literature, helping students kind of figure out how would they answer the question, what the heck is synthetic biology anyway? The second main goal was to have them engage in an authentic synthetic biology research experience. When I first conceptualized this class, I had NSF funding, and so all the students were participating in projects that were part of that NSF grant. And then the third was to develop proficiency and breadth in communicating scientific research. So they did this by learning how to keep digital lab notebooks, participating in journal clubs, writing and reviewing a short grant proposal, and learning how to design and deliver a research poster presentation. So the general kind of format that I chose to use for this class, it was going to be pretty research intensive, and I really wanted students to be able to be in the lab twice a week. Normally, our courses are formatted that we can have a lab in a 1-time slot a week, and then there are lecture time slots. So I figured out a way to make this fit into 2 lab time slots, but one of those time slots, I always teach it in the evening so that it doesn't interfere with students' ability to schedule other lab courses that they need. So I teach 1 evening time slot a week and then 1 traditional afternoon lab time slot. And my colleagues and I, sometimes we call this a clabinar because it's kind of a mixed class lab seminar that meets twice a week, right? And we're doing lab work, we're doing group activities, we're doing journal clubs in every class period. And typically I've had anywhere between sort of 8 to 16 students in this class. They're usually all junior to senior level life sciences majors here at Whitman. So I briefly kind of already mentioned some of the assessments, but one of the sort of main ones is an NSF style research grant and panel. And so I have them go through a mock grant panel and they learn how peer review works, et cetera. And so the sort of general outline of how I've set up the course over the 15 weeks of the semester is we have sort of topic areas building in complexity, sort of starting with an introduction to synthetic biology and to their research project, and then sort of building up in levels of complexity. And then the research project stage, I can talk a little bit more about this later if folks are curious, but a big part of it is needing to design, build, and confirm DNA pieces. And I found that takes a good 8 to 9 weeks to kind of get to that point. So that's where we really heavily invest our time. And then I have the students build yeast strains with those parts. And then the last couple weeks of class are spent doing functional tests with fluorescence assays. So we do microscopy, we do flow cytometry, and then the last week is their poster presentations. Okay, so just real briefly, like an example of the kinds of projects that I've had students do in this format. So the work that I've been doing in my time here at Whitman has focused on functionally annotating nuclear auxin signaling components from a variety of plant species. And to do this, utilizing this auxin bio and yeast tool that was developed by the Nemhauser and Clavin labs at the University of Washington, and that's where I did a postdoc at. And so you can basically express the auxin co-receptor proteins, the auxin receptors and repressors. When auxin enters into the cell, it leads to ubiquitination and degradation of the repressor. So if you have a fluorescent tag on that repressor, you can read out sensitivity to auxin by watching that fluorescent signal go away. And a lot of the projects that I've had the students work on are really focused on these repressor proteins. And it seems like they're kind of a tuning knob point in the signaling pathway. Some degrade fast, some degrade slow, and that kind of sets up the timing of how quickly an auxin response is initiated. So this is a really great tool to test. Okay, we have some genome sequence data. It looks like this gene encodes an auxin repressor protein. Does it? And so for the first few years of teaching this class is when I was part of an NSF funded collaboration working in maize. And so we looked at 16 different maize auxin repressor proteins, expressed them in the yeast system, and confirmed that they were functional as auxin repressors. And so a lot of what would happen, right, because the class, we get to do maybe 1 or 2 experiments by the end of the semester once we've built these strains. So oftentimes what I do is that's our pilot experiment, and then I'll have a summer student sort of pick up with that and do the much more careful experimental work with those repressors. And this actually led to a publication in 2020. And then in more recent years, I have a collaboration with a colleague at the USDA, Jessica Waite, who works on pears here in Washington State. And so the students in the Syn-Cell Bio class and I have been testing auxin repressor proteins from pear. And pear has a somewhat recently annotated genome. And so we've been helping to sort of functionally characterize some of these with hopes of having some pilot data for a grant proposal. So again, the students get to do typically confocal microscopy to look at where the proteins are being expressed in the yeast cells. And then we use flow cytometry to measure fluorescence over time in response to auxin to look at whether those repressors are degrading when exposed to the hormone. Okay, last little bit here. So I'll just talk a little bit about some of the successes and challenges I've had. So student evals are always really positive for this class. I think they just really love engaging in an authentic research experience. So they leave comments along the lines of, right, loved learning by doing. The lab work, grant proposal, poster presentation were fantastic opportunities to build skills as scientists. love to gain more hands-on experience in the lab setting and being forced to think analytically. So kind of to summarize what I've sort of seen in the responses to students, they really like that firsthand glimpse into doing science and being a scientist. Some of them have had previous research experiences. A lot of them haven't. This might be the first time they're really having an extended research experience. They love seeing a project go from start to end. They really like getting to grapple with failure and figure out how to overcome failure and troubleshoot in the lab, and the exposure to interdisciplinary science. For me as an instructor, I've found that it has led to, I'm really deeply intellectually engaged in the work, and I think that influences the student experience as well. It's been a place that I've been able to pilot new experiments and sort of new lines of inquiry that often become new projects for summer students to work on. And some of the stumbles along the way. Time commitment. I have let this class in the past really suck a lot of my time. And so it's been over the years a sort of challenge to figure out how do I balance the time that this class needs, the time that students are spending on it, right, and the time that I'm spending on it. So it requires some upfront experimental design and testing ahead of time, right, to make sure that the project is going to work out, likely work out how you expect. Prep work both before and during the semester, right, depending on the project, can be intensive. It's less so now because I've obviously done it several times, so I have a pretty good handle on what it takes. But those first couple times through, it definitely was. Setting up mock grant panels and preparing reviews is a pretty intensive process. I've played around with the number of journal clubs also, right, because that can be a lot of work of reading new papers. I always meet with the presenters ahead of time to kind of talk through the paper together, that sort of thing. And another bit has been data management, right? So this is can be like having 16 independent students working simultaneously on a project. So I've had to get really good about how do I keep and organize all of the primers they make, the plasmids they build, the bacteria strains, the yeast strains, ensuring that we stock stuff along the way so things don't get forgotten or missed. And then I've collected all student data and notebook in Google. So we use Google Docs, Google Sheets, Google Drive pretty extensively to collect everything. So it's lots of trial and error to kind of figure out the right balance of student and instructor workload. Okay, I'm going to stop talking there. I'll remind everybody about the ways that you can support the PUI section of ASPB. Only a $5 annual membership fee. And I'm going to go ahead and stop recording at this point, but I did put up a couple discussion questions that we might use as a sort of Kickstarter to talk about this a little bit more. So let me stop recording